Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore a fascinating topic known as chromosome mapping. Imagine trying to find a specific house in a massive, sprawling city without a map, street signs, or a GPS device. It would be nearly impossible. For decades, geneticists faced this exact problem when trying to locate specific genes within the vast landscape of our DNA. In this session, we will learn how scientists solved this puzzle by creating genetic maps, allowing us to find the exact addresses of genes and understand how traits are inherited together. To understand how we map these genetic addresses, let us start with a simple analogy. Imagine you are driving down a long, straight highway that has several small towns scattered along the way. If two towns are very close together, the chances of your car breaking down exactly between them is quite small. However, if two towns are hundreds of miles apart, the probability of a breakdown happening somewhere between them is much higher. In genetics, a chromosome is exactly like that long highway, and the genes are the towns located along it. By looking at how often genetic material breaks and swaps between two genes, we can accurately map their locations. Now that we have a mental picture of genes on a highway, let us look at the actual biology behind this. Formally, chromosome mapping is known as genetic linkage mapping. This brilliant concept was pioneered by Alfred Sturtevant, a student of the famous geneticist Thomas Hunt Morgan. Sturtevant realized that genes are physically linked together on chromosomes. During the cellular process of meiosis, homologous chromosomes pair up and can exchange pieces in a phenomenon called crossing over. Sturtevant proposed a groundbreaking idea. The frequency of this crossing over between two genes is directly proportional to the physical distance separating them. The further apart they are, the more likely they are to cross over. Understanding this biological mechanism naturally leads us to ask why we need to know the exact location of genes in the first place. In the real world, chromosome mapping is not just an academic exercise. It is a foundational tool used by medical researchers. It allows scientists to hunt down the genetic causes of severe inherited diseases. For example, before we had modern DNA sequencing, scientists used linkage mapping to find the exact location of the gene responsible for cystic fibrosis, which eventually led to targeted treatments. Furthermore, in agriculture, plant breeders use these maps to combine desirable traits, like drought resistance and high yield, into a single supercrop. Since we know that distance affects crossing over, we need a reliable way to measure it mathematically. Because we can't easily use a tiny physical ruler inside a microscopic cell, we use a mathematical proxy called recombination frequency. This formula calculates the percentage of offspring that inherited a mixed-up or recombinant chromosome. The variable R represents the babies that show a new combination of traits not seen in either of the original parents. The variable T is simple every single baby produced in that specific genetic cross. We divide R by T and multiply by 100 to get a clean percentage. When the physical distance between genes increases, the number of recombinants increases, which naturally drives the recombination frequency value up. 1% of recombination equals 1 map unit, also called a centimorgan. Now that we have our formula, let us see how this calculation works in practice with a classic genetic cross involving fruit flies. Suppose we are studying two genes, one for body color and one for wing shape. We are given that the cross produces 800 flies with the original parental traits and 200 flies with new recombinant traits. We are asked to find the genetic distance between these two genes. First, we find the total population by adding the 800 parental flies to the 200 recombinant flies, giving us 1,000 total flies. We divide the 200 recombinants by the 1,000 total flies to get 0.20. Multiplying by 100 gives a final recombination frequency of 20%. Physically, this means the genes are 20 map units apart.
Having successfully calculated the distance between two genes, we can now tackle a slightly more complex puzzle: figuring out the exact order of 3 different genes on a single chromosome. Imagine we have 3 hypothetical genes, which we will call gene A, gene B, and gene C. Through previous breeding experiments, we have already calculated the pairwise distances between each combination of these genes. Our goal now is to use those individual distances to draw a logical map that places these 3 genes in the correct linear sequence. This is a crucial step in building comprehensive maps of entire chromosomes. To solve this puzzle, we rely on a fundamental mathematical concept known as the additive property of distances. The strategy for this type of problem is to always start by identifying the 2 genes that are the furthest apart. The formula we use is simple. The maximum distance, denoted as D max, observed between any 2 genes represents the outer boundaries of our genetic map. Once we identify those 2 boundary genes, which we call B and C here, we know for a fact that the third, remaining gene must be located somewhere in the middle between them. If the physical distance between the outer genes increases, the map simply gets longer, but the logic of finding the boundaries first remains exactly the same. Let us apply this additive strategy to our specific experimental data. We're given the following pairwise distances. The distance between A and B is 5 map units. The distance between B and C is 15 map units, and the distance between A and C is 10 map units. We are asked to find the outer boundaries of our map. Looking at our data, we compare the numbers 5, 15, and 10. The largest distance is clearly between gene B and gene C, which is 15 map units. This critical piece of information tells us that B and C must be located on the extreme outer ends of our genetic map. Physically, this means they are the furthest apart on the chromosome. Now that we have identified our outer boundaries, we can begin proving the exact linear sequence of all 3 genes. This chunk of our logical proof is setting out to establish the relative position of the middle gene. Because genes B and C form the boundaries of our 15 map unit segment, the remaining gene, which is gene A, cannot be on the outside. It must be located somewhere in the middle between them. Therefore, our proposed linear order is B, followed by A, followed by C. The intuition here is just like points on a line segment. If you know the endpoints, the third point has to fall inside that segment. To finalize our proof, we must verify that our proposed order mathematically holds up. We check the math by adding the 2 smaller segments together. We take the distance from B to A, which is 5 map units, and add it to the distance from A to C, which is 10 map units. 5 plus 10 equals exactly 15. This perfectly matches the total measured distance between the outer genes, B and C. Because the sum of the parts equals the whole, we have mathematically proven that the correct linear order of the genes on the chromosome is indeed B, then A, then C. This simple additive logic allowed early geneticists to map entire chromosomes. With our proof complete, Let us look at a visual representation of what we just calculated. On screen, you're seeing a classic three-point genetic map. The long horizontal line represents a segment of the chromosome. Notice the 3 vertical tick marks representing the exact locations of our genes. On the far left is gene B, and on the far right is gene C. The large bracket at the bottom shows the total distance of 15 map units between them. Inside that segment, gene A is placed closer to B. The top brackets clearly show the 5 map unit distance between B and A, and the 10 map unit distance between A and C. This diagram perfectly illustrates the physical reality of our mathematical calculations. To wrap up our lecture, let us review the most important ideas we have covered today. First, chromosome mapping is a brilliant method that uses the natural biological process of crossing over to measure invisible genetic distances. Second, by calculating the recombination frequency, we can determine exactly how far apart genes are, translating that percentage directly into map units. Finally, by applying the simple additive property of distances, we can piece together the exact linear order of multiple genes. You have now mastered the foundational key to understanding how traits are linked together and how genetic diseases are tracked across generations. Great job!